All right, why don't we go ahead and get going? So I just want to say welcome to everyone. My name is Sarah Samuel. I'm an informationist at the Taubman Health Sciences Library, and I'm also a member of the Research Data Stewardship Initiative's working group, which is why I'm moderating the session today. In case you haven't already heard about us, I'm sure some of you have probably heard us in passing, but the Research Data Stewardship Initiative, or RDSI, helps U of M researchers improve the transparency, rigor, and impact of their research and scholarship across all disciplines by providing resources and guidance around data management. If you haven't already, I encourage you to check out the RDSI website for more information about the initiative. And we have a variety of resources available for folks all around data stewardship on our website as well. And I think, yep, there's a link that was stuck into the chat. Thank you so much for that, Lindsay. All right, so we are so glad that you could all join us for the second session of our winter seminar series, in which we are hearing from a couple U of M faculty who have expertise on data management and sharing. So during our time today, I encourage you to add your questions into the chat box. And then you'll also have the opportunity to raise your hand if you'd like to ask a question during our designated Q&A time, which will be after each of the presentations. So our first speaker today is Pam Davis-Kean. Dr. Davis-Kean's research focuses on the various pathways that the socioeconomic status, or SES, of parents relates to the cognitive and achievement outcomes, particularly mathematics, of their children. Her primary focus is on parental educational attainment and how it can influence the development of the home environment throughout childhood, adolescence, and the transition to adulthood. Davis-Kean is also a research professor at the Institute for Social Research, where she is the founding director of the Population, Neurodevelopment, and Genetics program. That's the PNG program. This collaboration examines the complex transactions of the brain, biology, and behavior as children and families develop across time and is representative of the population. She has expertise in quantitative methods, data science, the use of secondary data for longitudinal analyses, and open science practices. Dr. Davis-Kean will be presenting on the open science pipeline, pre-registration to data sharing. So I'll go ahead and stop sharing my screen and turn it over to her to get started.
Thank you so much. Let me make sure, let's see, do my share screen. There. So as usual, I have to ask, can people see my screen? Great, thanks.
Yes, looks good.
Okay, so that's going forward. All right, so today, so first I'm just going to do my thank yous in case I don't get to the end. So thanks for the Open Science Framework, because they have this wonderful archive of presentations that I can pull from to create new presentations, which I'm doing today. I'm not going to cover, I do a full kind of pre-registration presentation, but today I'm just going to kind of go over why you would do it. And then I want to actually show, a lot of people don't know what it looks like when you've kind of completed a pre-registration all the way through to data sharing. So I wanted to show that. And then my lab at the University of Michigan Human Development and Quantitative Methods Lab, who's been on this journey with me as we've moved all of our kind of procedures to more open science procedures. They unfortunately have to kind of follow what I tell them to do, and they've been very good at it. And we've learned a lot along the way. All right, so I'm just for a moment, I also, this year, I'm working at the National Science Foundation as a program officer. And I just really briefly wanted to put this out that in August of last year, we had a decree come down from OSTP, which kind of governs what we do in the science agencies, basically saying the White House wants us to make sure our research is available without delay. No embargo, all embargoes released in order to have our articles available and also our data available to the public as soon as possible and at no additional cost to the American public. So right now, NIH and NSF and other agencies, DOD, other groups are putting together this open access policy that will go into place in 2025, no later than 2025. Some are going to be earlier. So I just wanted to kind of put that out as this background of why all of us are beginning to talk about these issues, because it will be the case that as you continue to look for grant funding, this will be what we have to do and provide to the community outside of scholars, so they have availability of our federally funded research products. That's all I'm going to say for my NSF side. I'm going to go back to my U of M side now. So one of the things I'm going to talk about today, and I think it's good to think about what's confirmatory versus exploratory analyses. And the reason I'm talking about this related to pre-registration is because a lot of people get upset with pre-registration because they believe that it sets you on a certain mode of which you can't change, which is not necessarily the case. But the reason why pre-registration is useful, especially for the social sciences, which I come from, is that a lot of our social sciences are kind of top down. They go from theory to hypotheses to collecting data and analyzing the data. Even if that's not what we've done, we often set our journal articles that we write or the papers that we write as if we are confirming something. And so pre-registration is particularly good for this context of confirmation, which comes from a lot of the social sciences. It's traditional hypothesis testing. Again, it's theory driven. We want to hold the results we get kind of to the highest rigor. Did what we think we hypothesize, did that actually happen? And the goal is to minimize false positives, which means that we find something significant when it's not significant and makes the p-values that we get interpretable. And then there's the exploratory, which is the contact of discovery. It pushes knowledge. It's often data-led. It can find unexpected relations that we didn't see because we constrain the data in this more confirmation one. And its goal is to minimize false positives, which is that we don't find something that's actually there in the data. The issue here is that the p-values can be sometimes meaningless in this situation because we're running so many tests that we're messing around with the probability of the p-values, where we might find something one time to be significant, another time not. So in the case of, I'm really going to be talking about confirmatory. You can also, in the case of pre-registration, say you're doing exploratory analyses. I can do a whole nother talk about how to think about that. You need to kind of charge yourself some extra things if you're doing exploratory so that you make sure that you're not finding unexpected relations that are untrue. But today, the pre-registration really works the best for confirmation. And so I'm going to talk mostly about that. But I wanted to make sure people understood that there's nothing about pre-registration that's trying to dictate to you how you look at your data. It's just trying to make sure that in situations, especially when you have competing theories or competing research out there, it's a really great way to put a lot of rigor on the top of something so that at the end, you feel comfortable that you've actually answered the question that you tried to answer. And that's really the goal of pre-registration, not for any group of scientists to kind of put down some rules on top of other scientists. So this is the hypodeductive model that we often use in the scientific method. And so I just wanted people to, sorry, I hope that's not showing up on the screen the way it is on mine. So I just want people to kind of look at what is generally how we do science. We generate and specify hypothesis, and we often have, at this point, in red, our failure. So at this point, we have a failure to control for bias, the bias that might come into our hypotheses. We design a study. If we don't have enough people or animals or any group that we're looking at, then we have low statistical power, which means we've already gone into our science, not getting enough statistical power to actually find whether or not the p-value will be significant or not. We conduct the study, but if on the way we're doing really poor data management, we're making decisions, we're changing the way that we've done the study, then we have poor kind of quality control. When we get to the analysis data and testing our hypotheses, we're constantly running tests that we then don't tell people necessarily that we're doing it, and it's p-hacking. So every time you go in and you pull a ball out of that kind of probability thing, you have a higher likelihood of finding something significant that actually isn't. And interpreting results, if they've been p-hacked, you suggest again, there's something there that's not there. Along the way, there's this thing called harking, which is hypothesizing after the researcher is known. So you've already run all the data. You didn't really have a hypothesis or theory. You found something, and then you walked it backwards. You actually wrote your paper backwards from the data, and you went and found something that fit your findings instead of your findings being related to the question that you had. And that's what harking is. You go backwards and pretend that this is how you always want it to be there from the findings of your data. And then we publish this if it's significant, because we have a publication bias towards significance. If you publish it, then this is what goes out. We have all these significant findings, whether they've been HARCed or p-hacked, coming into our publications, and those publications are out there what we know about our science. So a lot of what open science is trying to do is making sure we are totally transparent about what we're doing, so that in the end, everyone can trust our science. That what we've done is use the scientific method correctly, the way that it's supposed to be done, so that people can be confident when they read our science that it's done correctly. So that's generally the goal. Again, not to try to dictate anybody's science, but to just make sure that when we do the science, people can trust that the science has been done well. So what are some of the problems that we have? I have to move things around my screen because lots of things are showing up on taking over my screen here. Okay, so I'm going to be highlighting these problems. They're all in this one sentence. This entire sentence has the 3 problems I'm going to talk about. The combination of strong bias towards statistical significant findings and flexibility in data analysis and results can lead to irreproducible research. So the first thing is a strong bias toward statistically significant findings. So I said this a little bit for publications. It's very, very hard to get non-statistical, positive findings or negative findings published. And it means that our entire set of publications are based on finding significance, even if significance wasn't there. And we still continue to have that strong bias in all of our publications, that we only think something's important if it's statistically significant. In my world, there's a lot of things that are important to find out if they're not statistically significant, because we don't want to keep doing that science. And so this is, again, kind of a problem if we keep going toward finding, if we do these issues, p-hacking, HARCing, we're putting things out there that keep us doing a science that may not have the rigor it should be doing. We're training students to do that science, and it may not continue to be something we should be doing. So in this world of the fact that science is supposed to work, where we find things significant, we continue to pursue those. Things we don't pursue those. We've kind of messed up that system, which means we could be continuing to do science that just really isn't relevant to the outcomes that we're interested in my case, to human behavior, but it's true for all of our science. So that's problem 1, the strong bias that we have in our publication record. Again, we can talk about issues of journals and publications, but for now, it's the bias we have towards statistical findings. Sorry, my thing won't move forward. Just a second. There we go. So number 2, flexibility in data analysis. So this again is the p-hacking, the HARCing, the p-hacking, and the management, the data management. So decisions that are made all along the way, and this is something pre-registration will help with, is that we make decisions and we make them on the fly. So you've got the data in front of you. I'm a data scientist. I do this all the time. You have a rich set of data. You can just really run that statistical analysis. You can answer your question right now. You don't have to wait. But by doing so, you're kind of messing around. Oh, now you have a little bit more information. And I turn to one of my students and say, isn't that cool? Maybe we should try this. Maybe we should break it out by groups. Maybe we should do this. Well, now we've started going down what Andrew Gelman calls, the forking paths, right? The garden of forking paths. Now we're really discovering the answer to a question that we didn't even have the question for. We just are playing with the data and we're finding something that looks interesting, but it wasn't where we really started. And so all of this flexibility means the kind of stuff that we're publishing may not actually be where we started. It wasn't the problem we started with. And we might be finding something totally different. And again, creating something and publishing something that's not even a true finding, but something that just showed up because we were sitting there just walking down, trying all these different things, putting in interactions, trying to do something in a different way because we were kind of exploring our data. This is where people start saying, oh, but it was exploratory. Perfectly fine, as long as you admit it was exploratory and not confirmatory. So again, this is one of our problems. Problem 3 is once we do all of that, if we don't have it all written down, people don't know what we've done, they can't follow our lab manuals or our lab books as we've written everything down, then we can't reproduce that research. And we have this very strong sense in our science that whatever we do, and this has been from time immemorial that I can remember in science, we're supposed to write everything down so that the next person can take our lab books and reproduce whatever we have found. And that's something that, at least in psychology, we've lost track of, our ability to really reproduce our research. And part of that is because we don't have the scripts that people have written in all kinds of different ways. What was your question? Why did you ask this question? Why is it important? How did you go about collecting the data? We just don't have that information down. And so because we don't have that information available, the next person that wants to try to find the same finding we have, they have nothing to fall on except what's in our journal articles. So that's another thing that pre-registration helps us with. It allows us to have the script of every decision made by the person who did the study, so that if we want to go reproduce that, we can go along and just read through their pre-registration on the same data set or on a different data set, depending on what we're trying to do or what kind of question we're trying to answer. All right, so what is pre-registration? It contains your hypotheses, your data collection procedures, your manipulated and measured variables, your statistical model, your inference criteria. So you put everything down, right? So what does this sound like to some of you out there? This is any paper that you've ever written. This is any study you've ever done. It's your master's thesis, it's your dissertation. You're supposed to put your hypotheses out. You're supposed to tell us how you're doing your data collection and your procedures, what your variables are, what your model is, and what you're going to take from that if you find what you think you're going to find is true. So again, this is nothing new. This is exactly the way we train every student to actually do science. And all we're asking is for people to actually put this all down before they collect any piece of data. So that's really what pre-registration is. This is writing it all down, just like you would do any theses that you put in, anything that you do ahead of time, write it all down before you touch the data. You should not be looking at the data when you're doing a pre-registration. And so then what does it help us with? Well, it's this kind of file drawer effect. So one of the things you've probably heard a lot of people talk about is the non-significant findings that we find often, and I'm going to say that often, are things that we don't publish. They're put to the side. And so what does that mean for our science, or what they call the file drawer or garbage? So what does that mean? We have all these null findings that we don't have a distribution on. All we have is the positive side of the distribution, and we have nothing on the negative side. So we have no idea how much of the findings we have might actually be pulled to more of a null side, because we don't have that null side anywhere that we can look at. So by allowing for pre-registration, allowing for things to be found not significant, it allows us to have the other part of the distribution, so that we can actually see, well, is the positive findings like just a really small outcome of mostly negative findings, right? So some people did find things to be true, but it was for certain populations. It wasn't for everybody. And we actually have more null findings than we have significant findings for this phenomena. Right now, we have no way of knowing that, because the null findings don't get published and they don't get put anywhere for us to look at that. So we continue to have this very positively skewed distribution. So people talk about, oh, you should do a meta-analysis. Well, meta-analyses are only on the things that are generally published. Some people try to go after things that haven't been published, but most of the people, when it hits their file drawer or their garbage can or wherever they put the null results, they can't find them for you because they just don't pay attention to them anymore. Sometimes they are things that are in master's theses and dissertations that people just let go. So we can't find them. So at this point, without having things pre-registered, without having an avenue for having null results like a preprint server would allow, we only know this positive distribution. So we don't really know how any of these phenomena we look at actually distribute across findings. So again, this is one of the things that if you pre-register and you get a null finding, and one of the things I'm not going to talk about today, but it's pretty common now, people are talking about, which are registered reports, which is actually the way to like get everything published that we want to get published. Then again, we're just kind of stuck here, but it's one of the things that at least could help fix it. It helps fix this flexibility in data analysis, or often what people call p-hacking, but there's other flexibility. If people do structural equation model, there's a ton of flexibility in ways you can manipulate the structural equation model to find things to be true. So there's other types of things you can do in this harking. So these were kind of the three things it was set up to fix. But there are other things that it can help with along the way. All right, so what problems, again, what does it fix? So it makes the distinction between confirmatory and exploratory, makes it clear. You set it all out. Presenting exploratory results as conformity for publishability, is at the expense of our credibility. So it's one of these things, again, where we're like, this is actual confirmatory hypothesis. And I will tell you from having done this, I just had a student do this for their master's thesis. When you have controversy in your field over competing theories and competing findings, pre-registration really helps you narrow in saying, this is what we're looking at. This is what the literature is telling us is going on. And because this is what it's saying, here is how I am registering the hypothesis to be very clear. And it holds it there so that when you finally have the outcome, you know you've like looked at all these other things. It's not because you messed with the data and found what other people were finding. It was because you're actually testing that theory. And it was very nice. She was in a situation with lots of competing theories that she was able to lay it all out, do the analyses as she pre-registered it, and get a finding of whether this theory was correct or this theory was correct. And that's just huge. It helps us move on and think about how to do things differently instead of just confirming what somebody else found, because we kept looking at the data till we could confirm what generally is already out in the literature. All right, and this is just how you would go about doing it. You would create a pre-registration, and this is kind of how you can help, like with exploratory. So you create a pre-registration, you collect new data, you confirm, but you can also at this point do discovery, right? So you confirm something, now you go and you look at the data and you're like, but I find something else. Well, now I'm going to go back and check that. I don't know if that's just like a fleeting thing or not. So now I'm going to go pre-register, and now I'm going to take it through the much more rigorous hypothesis testing of pre-reg to see if this thing that I explored and found actually is true. So this is, again, the confirming stage that you can use for exploratory data analysis. You went, you found something really interesting. You want to show that it's not just a relation that you found in your data. Well, now you're going to go back through and use pre-registration as a way to really solidify those findings. Check my time real fast. Okay. I just want to let you know that on the Open Science Framework, there's a whole host of pre-registration templates, because everyone's like, how do I pre-register? There's all kinds of them. I worked on one that's called the secondary data pre-registration. So you can pre-register secondary data. What that means is that you don't look at the data set. You don't download the data set until you actually do your pre-registration. So you have not looked at the data. It's equivalent of having not collected the data before you actually do that. So again, all of these pre-registration forms and templates are available. Almost every area has made one. So you can find them. And if it's not there, you can make your own. And you can put it through and have OSF look at it and see if it meets the criteria of a good pre-registration template. Again, it's totally open to the community to do this. And there's ones that are all like fully complete. I will tell you this one that's called As Predicted is it's only 8 questions you have to answer for pre-registration. So it's pretty easy to do. And these are there's qualitative, there's cognitive modeling, there's fMRI. Again, and this was from a while ago. So there's even more pre-registrations up. Okay, so what do you need to think about for your research group? And I just wanted to do this very quickly before I go to the example of what the pre-reg actually looks like. You want to talk to them about preventing p-hacking and harking. You want to create a standard procedure for pre-registration, both for novel research and secondary data. You want to talk to them about the extent data and code sharing is encouraged within your research group and outside of your research group. These are things that just come up all the time when you're talking to people about how to move them to this more rigorous one. What is your dissemination strategy? In our lab, everyone posts to a preprint server. Sometimes we have to deal with the journals on how they do that. But now, and because this public access thing is coming in from Washington, preprint servers are going to be the way that we need people to disseminate their findings if they're not paying open access fees. What are the standards for your material and code being made open? Where and how should research group members be expected to make it open? What are the kind of archives? So these are the ones I use: ICPSR, OSF. PAR is the NSF archive. I was speaking about this before we got on. The National Science Foundation actually has their own archive. I did not know about that till I started working there. And of course, NIH has multiple archives. Some of them are demanded. If you do certain kinds of work, you have to archive at NIH. Other ones are voluntary for archiving. All right, so I'm going to stop sharing this screen, and I'm going to show you what it actually looks like. Let's see. I'm going to share an OSF. one. So a lot of people say, well, I don't even know what this looks like. How do I go about doing it? So I wanted to show you this most recent paper we did. So the paper was called Charting Functional Forms of Short-Term and Working Memory, 2 nationally represented samples. This is what the preprint looks like. And what you can see here that on the preprint, which is the paper prior to publication, this is published in Developmental Psych, you can see that there was a pre-registration available. You can click to the pre-registration, and you can see that we made, in this case, we don't own this data. This is publicly available data, but we gave the HTTP things to go get it. So if you want to ever replicate, this is where we got our data. This is where you can go get the data. Again, we don't own the data, so all we can do is tell you, here's the data to go to. But one of the things we have on the registry, so here's where we did the data collection. Here were our hypotheses that we set up along the way. You can see all the things I talked to you about. This is as predicted, which is 8 questions. So you can see how short it is. We had to talk about outliers and exclusions. We had to give our sample sizes. And then there were other things that we were doing as well to have in here as what we had. On the side, you will see that there's also information about other resources. We did share our analytic code. We do have supplemental data that we made available, because once you do all of this, you end up with a ton of extra just stuff you want people to see that you couldn't put in the paper. And so we have that associated with it. This is kind of in the registries and the project. So it all ends up being associated with a project in OSF. Let me see if this one, yes. So this is what the project looks like. You can see everyone's name. We have the syntax available. Everything of this paper is open and available for anybody who wants to look at it. And it's all within the OSF framework, all the way to the preprint. You can find, and when you put that preprint up, you get a DOI. And that's really useful because people can start citing you even prior to that. And then on Google Scholar, you can actually merge your preprint DOI with your published DOI. So at no point in time do you end up kind of with these 2 different ones. You can always put them together, and they can always be findable even prior to the time that they're published. But you can see that everything that we have that anybody needs can be found because we've stored it all of our syntax, all of the supplemental data. And just briefly, I only have a few minutes left. I do want to say, and I'm going to go back to the OSF registry. We had to update our registry 4 times. And put in supplemental information because this was secondary data. Things that we expected from reading the code book when we actually downloaded the data was not the case. And so at that point, we had to stop everything because, and again, we had the data in front of us. We found it when we were doing the data. Our tendency was to say, oh, just do this. This is what we've decided to do. But Alexa Ellis, who was one of the researchers on this, said, no, we can't do anything that go against our pre-registration. We all have to stop. We have to decide what our next step is going to be. And then we have to put that in as a supplement. And that ended up being a really kind of wonderful addition to have, because you can see 4 times we had to deviate from our pre-registration because the data didn't end up being what we thought it was going to be. And that's totally allowable. You just have to make that decision before you actually run the data. And in the end, all of us at the end of this felt like this was the strongest, most rigorous paper we had ever written because we held ourselves kind of to the highest amount of the pre-registration. So we felt very comfortable in the findings that we had at the end. There was no kind of messing with anything or doing anything that we don't have listed out. And so that was an incredibly positive experience. And I think you could ask any of the authors on here and they would say, wow, that was probably the most fun and most interesting part of science that I've done. So I'm going to stop sharing and I'm happy to answer questions. I think have some time.
Awesome. Great. Thank you so much. Yeah, we just have time for like one quick question before I want to make sure that we get our other speaker in as well. But we did have a question come in about when is pre-registration supposed to be submitted? How does one contend with delaying submitted pre-registration to avoid being scooped? Or can pre-registration be considered intellectual output on par with publication? And this was a question submitted in the chat if you wanted to see the text.
Yeah, because there were like 3 pieces earlier to that.
Yeah, it was a little earlier up in the chat.
Okay, so yeah, let me push up. Sorry, get to the top here. All right, when is pre-registration supposed to be submitted? You're supposed to submit you're supposed to do a pre-registration prior to collecting data or looking at the data set if you're using secondary data. So you should not have looked at the data except for a code book to do a pre-registration. We always submit our pre-registrations as soon as we feel like we've covered everything that we want. Why do we do that? Well, this kind of goes to the issue of being scooped, though I don't think people are scooped as often as people think they are. We get our pre-registration up and we let people know we're doing that science. The reason why I don't know that it helps is because that means people actually did a search to find that we had a pre-registration up, but we have a timestamp, and we have a timestamp on OSF or any other pre-registration site that says, this is when we decided to do it. This is when we decided to make these choices, and it's public and it's known. You can put up a pre-registration anonymously. Some people do that when they have things in for grants, when they are having things under review. We had to do that at one point for our pre-registration. We actually had, during the review process, we had to make it anonymous so that reviewers wouldn't go and try to look and find out who we were. This is this whole anonymous review by pre-registration, but you actually can do that on open science so that they can't find that. And can it be considered intellectual output on par with a publication? Good question. I think it is our lab notebook, which is part of what we do. I'm like I said, I'm currently at the National Science Foundation as a program officer. This is something I would be perfectly fine with people putting in the product when you have to say, what did you do this year? You may not have collected data, but you may have created multiple pre-registrations for the articles you're planning on doing. That would show activity for that year, even if you haven't collected data. And so in that way, it is a product, and it is a product that we would actually accept as being work that you've done on the project. And in fact, I'd much rather see those pre-registrations come in first than publications coming in before I know you've even had a chance to collect your data. So yes, in that way, it is intellectual output. And it, again, as I showed you with OSF, it's all interconnected as a project. And that entire thing is one intellectual output, of which the journal article is just the final stage of that entire scientific journey.
Awesome. Thank you so much. We're actually going to have to move on. I want to make sure we fit in our second presentation, but we can address some more questions during the Q&A time later. But I do want to introduce our second presenter is Pat Schloss. Dr. Schloss is the Frederick G. Novy Collegiate Professor for Microbiome Research in the Department of Microbiology and Immunology. His research group studies the role of the gut microbiota in human health and disease using DNA sequencing and reproducible data science practices. He is active on campus as the sponsor of a local chapter of the Carpentries, through which he and others introduce U of M researchers to implement reproducible research skills. Since the beginning of the pandemic, Schloss has been posting videos to YouTube teaching these skills to a broader audience. His Rifamona's channel has nearly 13,000 subscribers and 265 videos. In 2022, Dr. Schloss was inducted as a fellow of the AAAS and received the ASM Microbiome Data Prize. Pat is also the chair of the journal's committee for the American Society for Microbiology, where he has helped to promote issues around DEI and open science. He received his BS and Ph.D. from Cornell University, where he studied engineering, and then did a postdoc at the University of Wisconsin. So we'll be hearing from Dr. Schloss about, if you want to go fast, go alone. If you want to go far, go together. Examples from engaging in open science. So please take it away.
Great, thank you. And so everything looks good? Great, okay.
Yes.
Well, and thanks to the organizers for giving me a slot to speak today. And I think a lot of what I say will dovetail nicely with what Pam talked about and what you've perhaps been hearing in other instances of this seminar series. And so I, as the introduction said, am coming to you from the human microbiome field. And so my lab is interested in the role that the bacteria that live on and in us play in maintaining human health, as well as how deviations from kind of the normal community composition of those bacteria drives to disease. And so this has been an area of research that's been hot, so to speak, for the past, I'd say, 20 years. And it largely builds off of using DNA sequencing-based approaches. And so I think as fields are concerned, DNA sequencing community has drunk the Kool-Aid, so to speak, on making data publicly accessible. But as we find when we kind of start bridging communities together, we kind of see some of those practices fall by the wayside. And we've certainly been seeing that in the microbiome field, where we start seeing papers have phrases popping up saying data available upon request. And so you start seeing editorials like this one from Langill et al. In the Microbiome Journal a few years ago. This is actually basically calling out an individual data set that's perhaps one of the largest data sets that's ever been published. If you want to get a hold of it, you've got to pay thousands of dollars to get the data. You've got to process it on their servers. It's just a whole bunch of hoops. So the data is open, but not really, right? It's more of a performative openness. And so I've been gratified to see the NIH data management and sharing policy and to hear things from NSF and other agencies. I think this is really good for science. I think open science is good for science. At the same time, I feel like these policies are a bit like building the airplane while it's in the air, to overuse a metaphor. Sure, like in the DNA sequence community, we have databases where I can put raw data, right? I can go to the sequence read archive, the SRA, to put my data. We're also starting to do metabolomics data. There isn't a government-based or stable database for uploading mass spec data, right? So if I want to upload mass spec data to make it publicly accessible, I'm going to upload it to researchers at UCSD. And if they lose funding, or if they retire, if they move, if they lose interest, all that could go away. And that sounds hyperbolic, but that's actually happened within the microbiome field for websites actually that were run by the Department of Energy. And so when we hear these stories about permanence of data, that's very real. It's not kind of slippery slope types of arguments. And so it's really nice to have these federal databases that are stable and will persist and that are open and secure. And so again, what I would say is, yeah, we can figure things out for raw sequence data. Other types of analytes like mass spec data are harder. And I also sometimes get rumblings that NIH isn't totally sure what they bought into when they started making all of the raw sequence data available and wondering whether or not they have enough resources to store all that data. And I think that's going to be a concern that comes up more frequently. It's like, yeah, we want to have open data, but can we actually afford to do this? And so the sequence data or the biological or chemical data isn't enough. What we really also need is the data about those data, right? So if you deposit a bunch of sequences to the SRA, it tells me nothing, right? I need to know, like, what kind of sample did that come from? Did it come from a person? Did it come from soil? Did it come from air, from water? Where, right? And so a number of years ago, a consortium of microbiome folks, including myself, got together and created a specification for the minimum information about marker gene sequences. And so this is the minimum metadata that we should be putting on every sequence. And I remember asking Frank Oliver Glockner, the last author on this, at a meeting, like, well, what is that? You know, what is the minimal information that you can say is required of every sequence across all environments? He's like, well, latitude, longitude, elevation. And as I got more and more involved in human research, I started thinking, well, no, actually, that's not possible, because I don't want you to have my latitude and longitude where I might produce a stool sample, right? Because that's where I live, right? There's security issues there. There's other issues. So we've gotten samples in the databases from the International Space Station as it's orbiting the Earth. And so if you look at its latitude, longitude, it's Kennedy Space Center down in Florida, right? And maybe its elevation is useful, but I don't know, right? And so we can very quickly kind of break down what is the minimum. And what I teach people or try to encourage people to do is be generous. Be generous with the metadata that you provide. The metadata might not be important for your personal study, but it will add value to your study if you provide more information, right? The more you can tell me about the patient, the field where you got the soil sample, the more value that's going to have down the road. I can't think of all the possible questions to ask of my data. And so again, if I put the data out there with extra metadata, then more creative people or differently creative people will come along and hopefully be able to answer those questions as well. Again, this has been kind of a hard nut to crack. And so there's been another working group that got together in this policy forum published in Science, talking about unrestricted use of public genomic data, that if you publish your data, if you put out the paper, then you should make the data available. To these issues of scooping, you've generally had probably, if you release the data at the time of publication or the time of review, you've probably already had the data for a few years. So you're already ahead of your competition by a few years. And so kind of to the question from the previous session, I think a lot of these concerns about scooping are a bit overbaked. And so one of the frustrations that just drives me nuts to no end is that we can't seem to get on the same team. And so this is the National Microbiome Data Collaborative, which I guess I shouldn't badmouth too much. They're the ones that sponsored the Microbiome Award I received. But if you go to their website, their NMDC team members, there's about 70 people working on this problem of trying to figure out how to make microbiome data more accessible. And what you'll notice is that of the 70, about 68 are from the national labs sponsored by the Department of Energy. Nobody is there from NIH. And so I feel like it's a real loss of an opportunity to bring together these federal agencies that are really leading the way in generating sequence data, microbiome data, to kind of figure out problems. And so then you potentially get duplication of resources and just all sorts of problems. And so this is clearly a complicated problem that I would like to think getting better at, but I'm not always so sure. And so if you kind of sit back at the lab bench, the reaction, I think, from a lot of PIs and a lot of microbiome scientists and scientists in general, and what I'm saying here is really, I think, illustrative of a lot of science, not just the microbiome, is that this seems like a lot of work. It seems really complicated. It doesn't seem always so easy. You've got to get the data in the right format. You've got to provide the right information. You've got to figure out how to upload the data. Perhaps that means using tools that you don't know how to use. And it's just a challenge, right? And so there's also, the perceived risks of getting scooped. And so I feel like so the researcher then finds themselves saying, why should I do this, right? Who benefits? How do I benefit? It seems like a lot of costs. And so there's all these great calls for making data accessible, but I feel like there's not a lot of good examples of how people have used effectively open access data to advance science, especially in the microbiome world and biological sciences in general. And so what I want to do is go through an example from my lab of where we have done this, right? Where instead of going fast alone, we went further together. And so one of the hallmark papers in the field of human microbiome research is this paper published by Peter Turnbow when he was a grad student in Jeff Gordon's lab at WashU. And so what they did was they got a bunch of twins from Missouri and sequenced their gut microbiota by a variety of different sequencing approaches. And the twins were concordant or discordant for obesity. And what they found was that there was a signal in the structure, the composition of their gut microbiota associated with obesity. So people who were obese tended to have a lower diversity of bacteria in their fecal samples. And they had a few other metrics that they were looking at. And so this became really a flashpoint for the microbiome field, that people's imaginations really latched onto this and like, oh, obesity is caused by the microbiome. And of course, ignoring the causation correlation conundrum. But you'd go to a microbiome talks or read books on the microbiome, Dr. Oz, whoever, right? The microbiome controls everything. And so we started wondering, like, well, is this actually true? Is this supported by other data sets besides the data that Turnbow and Gordon were looking at? And so a former postdoc in my lab, Mark Z, and I designed a study where we said, well, let's look through the literature and find all the data sets out there, sequencing data sets out there that we could find that would allow us to ascertain the obesity status of all of the subjects. And then let's go back and revisit the observations that Turnbow made, right? So this is kind of like the confirmation type of analysis that Pam was talking about. And so what we kind of re-ascertained this question of a link between obesity and the microbiome. And something else we wanted to do was that there'd been discussion on Twitter of people talking about conferences where they were saying you could figure out if someone was obese based on their microbiome. And so we asked if that's actually true, right? So could I take data from Turnbow, design a machine learning model based on Turnbow, and take that model and apply it to data from other data sets and correctly determine whether or not somebody was obese? So these are the kind of questions that we had that we were able to answer because we had a bunch of data sets. And so Mark kind of went through the literature and found 10 data sets that included large collections, and the gene we sequenced isn't super relevant to the general audience, but it's called the 16S ribosomal RNA gene. For this study, all but one of the data sets was available in a government type database. The one that wasn't was actually from Turnbaugh. And while Pam was talking, I went and looked, and it's actually still available on the Gordon Lab website all these years later. I suspect they didn't put it into GenBank at the time, or the SRA, because it was just nearly impossible to upload thousands of sequences at that time. So one of the messages also is that everything here is a moving target, right? Our practices, our beliefs, our technology is always moving. For the metadata, a good amount of the data from these studies was available in the Sequence Read Archive. Some of the metadata we had to get from the authors. We also found that some of the metadata was stored in dbGaP, which is a protected environment at the NIH where you need to write an IRB protocol, have that approved to get access to the data. For the data sets that we're looking at, I feel like that requirement was largely a roadblock to slow people down from actually using the data, or to keep people from using the data. If you've put together an IRB protocol, you know that it's not trivial, and it's certainly not as easy as downloading the metadata with the sequence data itself. There's no identifiable information in any of these data sets. And so again, I feel like, sure, the data is publicly accessible, but that might be more performative in the end. Also, there's a lot of heterogeneity to the data. Like, microbiome data is not generic, right? That there's different sequencing technologies that's evolved over time. Each researcher has their different part of the gene that we sequence that they're interested in. They all have their different protocols for processing the data. So this is a very heterogeneous problem. And what we really wanted to do is take all this data and run it through a common pipeline, analyze each of the 10 data sets separately, and then statistically pool the results to see if we can confirm or refute what Turnbaugh had seen. And so here's one of two slides of data, so just bear with me. I can't help myself. But on the left, you see the diversity. Again, in obese individuals, diversity, according to Turnbaugh, was supposed to be lower than in non-obese individuals. So Turnbaugh is about halfway down there on the left. And so we saw that result, albeit not a statistically significant result. And so of the 10 studies, we found 2 of the studies statistically supported that assertion, one from Baxter, which was from my lab, another from Goodrich. And so the interesting thing about many of these studies is that they really had most of them had no interest in obesity per se. So the Baxter data set and Schubert were from my lab. Baxter was looking at colon cancer, and Schubert was looking at C. difficile infections. And so again, because my lab and these other labs are generous in making their metadata accessible, we were able to take their data and kind of increase their longevity, right? And make them useful for other types of questions. I don't think anyone would say that we scooped these data sets or these investigators by using their data in our studies, since this wasn't really something they were interested in at the beginning. Overall, when we looked at diversity, at least, there was a very small but significant difference. I don't think it's a biologically meaningful difference in diversity, but again, because we could take these 10 data sets and pool the results statistically, we could then get a better answer than we could with any one study alone. Again, another thing we did was to generate machine learning models. And so in this slide, if you take that first column of Baxter, again, coming from my lab, the Baxter model was a random forest model where we took the point there with the open black circle, built a model to predict whether or not somebody was obese or not. And then we took that model and applied it to the 9 other data sets. And so as you can see, that hashed line basically indicates a 50-50 chance of being right. So basically random. And so basically our models were really bad at predicting obesity status in another data set, much less the data itself. And so we could replicate this then across those 10 data sets. Again, something we couldn't do if these 9 other investigators hadn't made their data publicly accessible. And so, as Pam discussed, one of the problems is the dreaded file drawer. the problem with the file drawer, right? Where if you have a negative result, it goes in the file drawer and is never seen again. And especially people are going to be very reluctant after the big splashy paper, like we saw from Turnbaugh. If you come along and you then contradict that, or say there's a negative result, then that's going to be far less likely to get published. And so the fear then is that there's lots of studies going into the file drawer that aren't getting published. And so one of the things that's nice to see about the NIH policy is that even if the data aren't published, the data still need to be made publicly accessible. Whether or not people will actually do that and how that will be enforced remains to be seen, but hopefully will further help to overcome this file drawer problem. One other thing that we did in this paper was that all of our code is available on GitHub. And so something to think about is code as a type of data. This repository is set up as a reproducible workflow. So it'll do everything from downloading the data to generating the final PDF. You can type make space write.paper at a prompt, and theoretically, it'll do everything for you. But as Mark points out in the README, it'll take you about 200 gigabytes of RAM and might take a few weeks to run. And so this is reproducible technically, but perhaps not practically. But again, the idea would be that just like we did these 10 studies, well, theoretically, we could come along again and add additional studies to see whether additional studies changes our result or perhaps gives us more confidence in the overall result. And so again, the title of this talk was, if you want to go fast, go alone. If you want to go far, go together. And I think the trend in science today, especially in the microbiome field, is to go fast, right? To have our splashy cohort, our splashy result, and then to put that out without looking at other data sets that might be available to see if it confirms or contradicts the result that we have. And so I think we need a cultural change in the microbiome field, at least, to go far, to go together. And so some of the things that I think we need to help make this possible are more use cases, right? So this is one case of where having that extra metadata, having the available sequence data improved our study. We need more people to do other types of use cases like these to justify to the reluctant scientist why they should be making their data accessible. I think we also need to separate out paper citations from data citations. I don't know that there's enough signal that a paper is getting cited because of the data versus because of the ideas in the paper. And so then I think we also then need to increase the value we place on data citations. And so finally, as an example of this, mentioned in the intro that I am the chair of the ASM Journals Committee. We have a journal within American Society for Microbiology called Microbiology Resource Announcements, which are 500-word papers to announce the availability of a data set. There's no results, there's no discussion. It's basically, here's the data. And so two journals, M Systems and Applied Environmental Microbiology, are working to pair resource announcements papers, data papers, with papers describing the results of analyzing those data sets. And so again, I think this is another cultural change, but something that we certainly need to help move open science and data accessibility forward. So with that, I'm sorry for going long, but I'd happy to take any questions people might have.
That was great. Thank you so much, Dr. Schloss, for your talk. So I didn't see any questions come in through the chat yet, but folks are also welcome to raise their hand if they'd like to ask a question. I'll be happy to call on you if you want to ask it audibly. Oh, Nick, go for it.
Hey, Pat, I was wondering if you could talk a little bit about the kind of the training stuff that you're involved with, because feels like there's a wealth of resources that, in addition to like what you just talked through, people can start to go spend time on their own. So like the carpentries, your YouTube channel, we could even post some links to some of that stuff here too.
Yeah, thanks for asking. Like that's become a real passion of mine. And kind of like Pam mentioned, I've led my lab through a journey over the last 10 years of engaging in reproducible data practices, trying to make our data more accessible, because in some ways I feel like the traditional lab notebook really doesn't work for what we're doing, that there's a lot of circuitous paths and a linear lab notebook just doesn't work. And so we've been working to develop programming skills. We use R, but it could just as easily be Python, version control with Git, literate programming with R Markdown, using things at the command line, high performance computer like Great Lakes. And so our lab is developing these skills ourselves, and people join the lab to develop these skills. And then we teach them through this Carpentries organization. So we teach 2-day workshops to expose people to reproducible research. And people in that aren't just from my lab, but from other labs as well. And what I find is that it gives me much greater confidence as a PI that we're engaging in these in the results if we're engaging in these reproducible practices with these reproducible tools, because I know that people are coding and they're doing their work like someone else is going to come through and look through their version control history, and so it just gives me greater confidence in that, and I think is pretty powerful. And then I guess the other thing I'll add to that is people have found it really useful outside of my lab. And I've had a number of just phenomenal bench scientists join my lab with no data science skills. They go through these training programs and they're now staff bioinformaticists at Merck when they join my lab with no data science skills. So it's exciting to see bench scientists develop these skills and to see the value of these skills and what they can do for their research.
Great, thanks. And I just wanted to highlight there were a couple links put into the chat, both to the YouTube channel and also to the U of M Carpentries website as well. So thanks for that question, Nick, and for sharing more about that, Dr. Schloss. Any other questions for Dr. Schloss? And while you all are thinking, I did just want to highlight, there was a question for Pam, Dr. Davis-Kean that came in just about, are you able to pre-register purely exploratory projects, or can you do it only for confirmatory? And Dr. Davis-Kean, did you want to give an audible answer to that? I know you typed an answer into.
Yeah, I put one in the chat, but they're absolutely right. It's hard with exploratory, but you do, what we generally do is, we want to test our models in lots of rigorous ways to make sure when we're doing exploratory analyses that we're just not getting a one-off finding. So it absolutely is the case that how you're going to go about doing that, what other data sets you're going to do. In the case, we sometimes use 10 to 20% of the data to explore on, and then we confirm on the other 80%, which is an absolutely legitimate way of thinking about looking at data. That can all be written down, just like anything else can be written down, so that people can follow what you did to get to the point. Again, these kind of pre-registrations, they're maps. They're telling people what you did so that they understand what underlies the science you had. But they're absolutely right. This has been a problem with data science in general because it's mostly exploratory, that we don't really have a set way. I'm not even sure we'd want to call it pre-registration, but a way of actually laying this all out so that people understand how we got to the end point of our data. So that is, if it doesn't exist, again, I invite anyone to create it. And I think, like Pat said, having these procedures more clearly down for everything we do is probably the best way to go forward, instead of it being just some group does it and some groups don't do it, right? We should have a standard.
Awesome. Well, thank you for answering that. And I did just want to say extra special thank you to both Dr. Davis-Kean and Dr. Schloss for sharing your time and expertise with us today. And thank you to all who joined us as well. I did just want to highlight that we do have another seminar coming up in April. So I encourage you to join us on April 7th for that. And you can find the link to register for that on our RDSI website. So if Lindsay, you can stick that link back so folks can get to that to register if they haven't already. So I just want to say thank you again. Hope you can join us, but then also, again, just emphasizing, we've got our resources and our RDSI website. Encourage you to check that out if you haven't already. So that's it for us today. And yeah, hope to see you all in April.